Hello, welcome back after, I don't know how long your night was, it was very nice. And we have a new session this morning, very interesting science. And first of all, I'd like to introduce Arwen Pearson, who studied in Bath and Bristol, and then, Bristol. And then went to the States, yeah, and was in Leeds for a long time, and then went to Hamburg, and built her own institute, which is quite achieved. Arwen is focusing on serial crystallography, on time-resolved work, and on multiple techniques. Arwen, thank you very much.
Thanks, Ivo, and thanks to the organisers for the opportunity to come. It's been a really great meeting so far. Good morning, everybody. Graham, I'm pleased to see you made it. Yeah, excellent. So I want to talk a little bit about serial data processing this morning, a little bit about the experiment, and hopefully set up where I think the challenges are that much brighter people, i.e. Elke, Nick, Ashwin, Bryony will tell you about solutions to, hopefully, and Helen as well, approaches to solutions to these later. So I thought we've talked a lot about single crystal rotation crystallography. We haven't really gone into what it's about. And Graham, I know this is boring, but I thought I'd put a little bit of revision in just to remind those of you who maybe haven't yet collected your own data, what's going on. So in single crystal rotation crystallography, we have a single crystal, we fire some X-rays at it, we collect a diffraction pattern, and then we rotate that crystal and collect a series of diffraction patterns. And doing the experiment this way means we know some things about our data, or we should know some things about our data. So we know, for example, the start and end angle of each image. We know how the images relate to each other in space. We know the order of the images, and we know that all the images come from the same crystal. Okay, and these are actually really important things when it comes to your data processing. We also make some assumptions, and you shouldn't forget those. We assume that the intensity of the symmetry-related reflections is the same, right? And that any changes we're seeing over the course of the data collection, as Elspeth showed you yesterday, are just a function, for example, of crystal thickness, potentially also radiation damage, beam decay, although that's really not such a problem with today's synchrotrons. We also assume that the structure is not changing during the data collection. That's not always a correct assumption, and that can cause you problems in your data processing if it's not true, but that's our starting assumption. And from this, we then want to extract some information. We want to extract the index of each reflection, so its position in the reciprocal lattice, and we want to associate that with an intensity for each reflection, or at least our best estimate of that true intensity from the available data. Okay, so this is what's going on when all that auto-processing is running, and what's happening in the background is you're indexing those reflections. You're then making a prediction based on that index of where every spot on each image should be, and you're going and doing an integration step, following that by the scaling and merging. And ultimately where we get to is an electron density map, and that electron density map should represent the average structure of all the molecules in the crystal. All right. What's also important to remember is that you can have maps that are very pretty and look very beautiful, but in the same map, is it going to go? There we go. You can also have regions that are very poorly ordered and poorly defined, because on average, the molecule is not in the same place, or that bit of the molecule is not in the same place in those regions of the map. That's something to have in the back of your head. So what's serial crystallography and how is it different? Right, so I'm going to ease us in gently. So we have what you can think of as multi-wedge data collection, and we've talked again about examples of that so far during the meeting. And here we're collecting small wedges of data from lots of crystals, all right? And again, we know some things. We still know within a wedge how those images are related to each other, okay? And we're usually, when we take a multi-crystal data set in this way, we're looking for the subset of wedges that best merge. And ideally, we'd like to have all the wedges in, but we're often looking for the subset that best merge. And I put best merge in quotation marks because people use different parameters to determine that best merging. Are you going by some kind of merging R factor? Are you going by a correlation coefficient? Are you trying to match things with the most similar unit cells? And there's a ton of clustering algorithms out there and approaches that try and do this for you in an efficient manner. And again, the assumption that underlies this is that the wedges we choose to merge came from crystals that are basically the same. All right, they have the same space group, the molecules in approximately the same place in each asymmetric unit. They're basically the same. All right. So true serial crystallography pushes that to its extreme. So now we're going to have single snapshots, no rotation, single snapshots from many, many crystals, right? And we get one shot. And that means we don't know a priori, before we start processing the data, we don't know how those images are related to each other. So this, with diffraction, is a solvable problem, okay? Because in principle, if we index those images, okay, we then know exactly where that image is slicing reciprocal space. All right, we know exactly which bit of reciprocal space we have on each image, so we can put the images into order to make a 3D data set. We still, much like the multi-wedge data collection, we're still looking for that subset of images that merge best. Okay, and in a serial crystallographic data set, again, we're making the assumption that the images we choose to combine are from crystals that are basically the same. And I want to sort of give a warning here. In serial crystallography, we often find very different crystals in the whole load of crystals that we looked at, and they can be very different space groups, very different diffracting quality, all right? And so being able to sort those things out apart from each other is one of the big challenges that we have. But again, the software is well developed, and this bit at least works pretty well as long as you have enough data. So what about our knowns and unknowns? So we, as I said, we don't necessarily know how the images relate to each other. In principle, once they're indexed, we can put them in order. But we have, again, certain extra challenges. And one is if we have crystals where the unit cell dimensions are very similar, right? And this doesn't occur in all crystal forms. There's certain crystal forms that are more prone to this, but this gives an extra layer of complexity in the data. And so, for example, if we have here, I've got a crystal here, that's what I'm pointing on somewhere. So Nick says, I can't point at the thing, I have to point nearby and hope the mouse comes down. Maybe, there we go, right? So we have a crystal, and these unit cell dimensions are slightly different, but not by a lot. And that means when we've just got a single image, it's not always clear, right? We don't always have enough information on a single image to really distinguish which possible arrangement of our unit cell axes we have. And we have what are called indexing ambiguities. And this gives problems if we have just two possibilities and we have enough data. Again, there's software, Tom writes partialator, for example, and ambigator, which go through and try and separate those two different indexing solutions and then make everything consistent. Once you get into more than that, then it starts to get complicated and challenging. All right, so what I'm hoping I'm conveying to you here is that this processing of these data is a bit different to normal data, but we have software tools to do it. What we then want, we've got them in order, hopefully, and we've solved our indexing ambiguities, and we need then again to get to our reasonable estimate of the true intensity of each reflection. And here now, the fact that these crystals are basically all independent measurements causes us some problems. So we have, for example, here a population of crystals. I've lost my mouse again. They're all in the same orientation. I've tried to show that with the coloring, but they're all slightly different sizes, okay? They might have different mosaicities, okay? They might be different quality. One might diffract much better than another. Maybe one got a bit squished in the sample delivery. And you add on top of that, okay, as Graham showed you yesterday, potentially you've also got different X-ray beam properties. So that shot-to-shot noise in both bandwidth and intensity can again mean that the estimate of I for a particular reflection that we're getting from this set of crystals, I've lost the mouse again, where's it gone here, is very different. And so how do we converge to that sort of true estimate that we want? And for this, we basically play a numbers game. And we measure data sets with huge multiplicity. And this is why when you look at serial data sets, you see data sets where people have merged thousands and thousands of images, and they're reporting multiplicities in the hundreds. Okay, and part of this is because we hope that by averaging enough things together, we begin to approach an estimate of the true mean. And so just to finish up this section as a sort of rule of thumb, if you have a well-behaved beamline, well-behaved detector, well-behaved X-ray source, all right, and this is our experience on T-REXX, you need about 5,000 diffraction patterns for that whole process of indexing, scaling, merging to be reasonable, all right, to be reasonably stable. If your X-ray source or detector is noisy, all right, so if you're at an XFEL where you've got shot noise going on using an integrating detector, there is noise in integrating detectors, they're not the same as the pixel array detectors, then you need more data, right, in order to be able to account and overcome the noise that's being introduced in your data set. And that can sometimes be considerably more data. You may need 50,000 patterns instead of just 5,000, okay? So I'm not going to say much more about the processing because I want to get a bit onto the experiments and a bit onto the downstream challenges that come from data like this. But if you want to know more about serial data processing, I can really recommend Tom White's website with Christabel. He has lots of lovely tutorials on there that will walk you through the steps of data processing, and also a lovely series of papers that are really describing what's going on under the hood in a serial data experiment or a serial crystallographic experiment. So why might you want to do this? Why might you want to come out of the comfort zone of our boring, routine, single crystal rotation experiments? And there's a couple of reasons. So one is radiation damage, right? And we've already talked about this as well for this meeting. So if you can't get a complete data set from a single crystal without radiation damage really corrupting your data to the point it's unusable, then a serial experiment is your option. And good examples of this that have been very fruitfully examined, for example, at the free electron lasers and membrane proteins, and also metalloproteins that are very susceptible to specific radiation damage to their metal centers, where you really you have to go to the serial diffract before destruction approach, where we come in with as many photons in a few femtoseconds as we would have at a synchrotron in a second, in order to be able to record data before the protein has a chance to respond and that damage actually has a chance to occur. So we're basically outrunning some of the chemical processes that come on the back of the initial radiation damage events. The other reason to think about doing serial is if you're interested in time-resolved experiments. And this is because if you have a system that's not easily reversible, right, that you can't easily reset back to the beginning, once you've started a reaction, you basically, to take another time point, you need to put a new sample on. And we call these non-state reversible experiments, and these are problematic because you can't just do this on a single crystal. You need lots of crystals, and this is where serial has really come into its own. So are time-resolved experiments in proteins or in crystals possible? And I just want to, we've again skirted around with some of the other talks and that we also saw in the Diamond user meeting about the issues of protein concentration, packing, accessibility. I just wanted to say a few words about this. So the concentration of protein in the crystals is pretty much that of protein in the cell. It's about 300 mg per mil. Your concentration of protein in your crystal is tens of millimolar. All right, it's a very high concentration. As you saw as well at the Diamond user meeting, lots of proteins retain their catalytic activity in the crystal. And we can actually, if we don't have a big conformational change that disrupts the lattice, proteins will stay crystalline during turnover. This is an experiment where we've added some ligand to the solution around the crystal, and you've watched that diffusion front come in. Elspeth, I know there is no scale bar. This crystal is about 800 microns long, okay? So you're seeing a slow diffusive process. And this process actually in this crystal is associated with quite a large degree of motion, which I can't get the mouse to come away. Anyway, it's associated with about a 20-degree domain rotation and actually a change in symmetry. And if you let this reaction continue on, the domain actually rotates back and you recover your tetragonal symmetry. So this was a really surprising example for me, that you could have such a big conformational change within your crystal and the crystal would still survive. So having said all of that, that serial is the way forward. If you want to do a time-resolved experiment, I need to give you a brief public service announcement, right? And that is that not all mechanistic experiments need to be time-resolved, okay? So there's a technique that's been around since the 1960s called trapping. And you can do this mechanistically, where we set up the sample, the buffer conditions, such that the reaction can't go beyond a certain point. We can also do it with cryogenic flash cooling, basically the equivalent of the freeze-quench approach in entomology. And as I say, the structural biologists have been doing this since the 1960s. The methods are really well established, and the cryotrapping works if you're looking at a relatively slow reaction that's happening on the order of many milliseconds, then you can be fast enough with a cryocooling to trap a particular species in your reaction. And I want to give a plug here for one of the new toys we have in Hamburg, which is called the Spit Robot, which has been developed by Pedro Marabi and Eike Schultz. And the Spit Robot is a freeze trapping or cryo-trapping robot that uses the llama technology that Pedro and Eike also developed to spit little bits of ligand onto your crystals, and then that's attached to a hydraulic plunger, and it's all nicely timed, and it will plunge your crystals into liquid nitrogen and hopefully trap you an interesting species. And possibly we'll see some of this at Ashwin's talk later, but if you want to know more about the Spit Robot, Caitlin's also here, who is also in Petram's group somewhere. I don't know if she's going to wave. She's not going to wave, but you can talk to any of us and you can come and use the instrument. So let's go back to time resolved. We also discussed a bit in this meeting the ideas about how fast you can go. And again, I just want to make sure we're all on the same page here. So how fast can you go, and how fast do you need to go? So how fast you need to go depends a little bit on what you want to look at, okay? So we've got time here along this bar, logarithmic going from seconds. The mouse hasn't disappeared, down to femtoseconds. Come back, mouse. No, hopeless, I'm useless at this, right, femtoseconds. So if you're interested in chemical steps, all right, bond breaking, bond formation, then we need to be down in the picosecond, femtosecond time regime. If what we're interested in, for example, is allosteric transitions, if I can get my mouse up there we go, then we're actually looking at something very different. There we're into the millisecond, microsecond time regime, and that's also the time regime for your slowest steps in enzyme catalysis. So you don't necessarily have to go to an XFEL and have the ultimate fastest resolution if what you're interested in are reaction steps that are occurring and structural processes that are occurring on much longer timescales. So what determines the time resolution you can achieve? And this is, again, something that I think sometimes people struggle with. It's not the detector. So the time resolution of the detector is irrelevant for your time-resolved experiment. It's determined by whichever is slowest of your reaction initiation or the time needed to record usable data, all right? And that's a function of the brightness of your X-ray source. So how fast can we go? At XFELs, you can, of course, go right down into the femtosecond time regime, and this is really where XFELs shine. We don't have any other structural tools that get us down to this time regime. People do experiments right up to the millisecond time regime on XFELs, but that's usually where we are really decoupling the reaction initiation and the X-ray experiment itself, for example, with a sample delivery approach like the tape drive, where we can move the samples through a reaction initiation environment through into the X-ray beam to get to these long time scales without throwing away lots of our photons by having to wait a millisecond before a next X-ray pulse, for example. At synchrotrons, we can actually go quite fast, and synchrotrons will get you quite happily all the way down into the hundreds of picosecond time regime. So, for example, if you go to the Lowy end stations, like ID9, you're down easily into the hundreds of picoseconds. And with the new microfocus sources that are coming on dedicated to time-resolved experiments, so for example, ID29 at ESRF, Micromax will soon be online, the planned KMX at Diamond, these are going to be able to get, because of the upgrades to the machines, to the synchrotrons, these are going to be able to get us into sort of 10 microsecond time resolution territory. So please don't think that synchrotrons are only useful for millisecond experiments. You can go much faster. So what else do you need to think about in your time-resolved experiment? You need to think about your sample delivery, and there's lots of ways. We use fixed targets a lot on T-REXX. You can do a direct injection. You often see this at the X-Files, either into a free-flowing jet or as a viscous jet. You have closed microfluidic sample delivery approaches, and this is that tape drive I mentioned, where we are writing droplets with crystals in onto a tape, and because this tape can be arbitrarily long as you keep it always nice and humid, you can have a reaction initiation happening quite a time before you probe with your X-ray. So you also need to think about your reaction initiation, and again, back to our timescale slide, if you want to go faster than milliseconds, we're really thinking about photoactivation. Okay, if your system you're looking at is slower, then we can do this by mixing. Now, both of these put constraints on your sample size, okay? And not all photoactivation methods are equal, right? So just because you're doing the experiment with a laser doesn't mean you'll automatically get to femtosecond time resolution. So if you've got your laser is exciting a photoisomerization or a direct photolysis, then yes, you can get down sub-picosecond. If you're using photoacids, those are sort of hundreds of picoseconds. So for a pH drop, temperature jumps with an IR laser are on the orders of nanosecond, because you have to let the heat that you've pumped into the water transfer to the protein. And your organic photo cages tend to be much slower, and the very fastest of them maybe just get us down into sort of nanosecond time regimes, but the majority of those are much slower. So thinking about how you set your stuff up is also important. So I want to say a quick few slides about T-REXX, and just to show how we've tried to make things a little easier for you. So for those who don't know where T-REXX is, this is the Science Campus Hamburg-Bahrenfeld in Hamburg. European X-Files starts on the west side of the campus and goes another 3.5 kilometers west. The PETRA ring is an old collider, and it's huge. It goes all the way around the site. Can't steer my mouse very well. And right in the very last end station hall of the Max von Laue Hall is T-REXX. So that's where we sit. So T-REXX is a beamline that's dedicated for time-resolved serial. Okay, we don't do rotation mode experiments at all. We've been running since 2019, and we have a very flexible sample environment. We're not a super hot X-ray beam, but it's enough that we can get easily in the millisecond time regime, and we touch for well-diffracting samples into the microseconds. And what's very nice on T-REXX, again, Pedram and Eicher have developed with Fridtjof Telkamp at the Max Planck, an environmental chamber that lets us actually do experiments from below 10 degrees up to 70 degrees Celsius with full humidity control. So we can start to ask questions about thermostability and really start to look and probe more deeply into reaction kinetics. We control T-REXX through a GUI. This is MX Cube, the old version of MX Cube. And so you set up your experiment here in the workflow, your queue. This is telling you about what the beam's doing. This, instead of a single crystal, you see a chip. This is your dosor score telling you about the resolution you're hitting. And then we have a hit map running under here. And this runs real time with the data collection. So each blue spot is when we've hit a crystal. So we see very quickly during the experiment how well the data is coming. We throw all of that straight away up into auto processing. So we run an auto processing pipeline for you to take you all the way to electron density. And we aim to have electron density available for you within an hour of a chip finishing. And I think that's now down to less than that, so that you get very quick feedback within your beam time as to whether your experiment is going in the right direction. Did anything happen? And we feed all that back to you in I/SPY-B. So again, you've got your information about the data collection, pictures of your chip instead of your crystals, the results of your data processing, and you can open your electron density in Ugly Mole and have a look at it. So all very lovely. And this is not my work alone. I basically make the tea. This is a big team. So we work closely with Thomas Schneider and his group at the EMBL, and Stefan Fiedler, who runs the EMBL instrumentation team. Niels in the back here for spectroscopy, Bryony, who you'll see later, and Godfrey, honorary T-REXX team members working on Hadamard. Marina, the Duchess of Detectors, makes the detector work beautifully. I didn't talk about Diana stuff. And Pedram and Eike and Fritjof have done a lot of the engineering, and Cian gets an honorable mention as being one of our power users. So this is a big team, and we wouldn't work without all of them. So I want to just, my last minute or two, quickly give a couple of slides to hopefully set up the talks you'll see afterwards. So we can get the data, the sources are all there, the automation is there, the auto processing is there, but when you have that data, what do you do with it? And I want to remind you that most of the time in a time-resolved experiment, you're measuring kinetics, not dynamics. You're watching changes in populations. As you move along in time, I've lost my mouse again, you're seeing intermediates appear and disappear. And that means that at any time point, you're seeing an electron density that's the sum of all of the different species that are in your crystal. And so we need to have data quality that's good enough that we can disentangle those different species and model them confidently. And this is non-trivial, and you're going to hear more about this as we go on. So if I finish up, I think our open challenges on the data processing side are dealing with heterogeneity in the crystal populations. We throw away data at the moment because the crystals are a bit different to all the other crystals, but they still contain information. And I think we should be getting better at pulling all of that back in into our final data set. We also have challenges in our map interpretation. How do we handle mixtures? How do we include complementary data? And really, we also need better models. A single PDB is not a good model for modeling a time-resolved data set. And I hope in Elke and Nick's talk, they'll take you some way towards answering some of those challenges. And then I think from Ashwin and Helen and Bryony later, you might hear a bit more. And I'll stop there. Thank you for your attention.
Thank you very much for this presentation. So well versed and so well done. Very good introduction for us. Any questions? Yes.
Gustavo Lima from Astex Pharmaceuticals. Could you elaborate a bit more how you can place your snapshot in the reciprocal space without the information about which points are getting in and out of the diffraction plane?
So you're asking how from an index we can place our individual snapshot in order with all the other snapshots,
Yeah,
is that right?
because I kind of understand how you would do that with the wedges, but not with the single snapshot.
So the wedges obviously is easy. With the single snapshot, you do actually have it. If you can index the diffraction pattern in principle, you know then exactly where that is in reciprocal space. If you have only very few spots, it starts to become very challenging. And there are algorithms that can take extremely weak data. So the EMC algorithm from Jenny Wehrman and Sol Gruner tries to go at that approach, but they take very different, it's no longer an indexing-based approach. I don't know enough about it. It's a much more statistical approach to trying to reconstruct reciprocal space from very sparse data images. So it is possible. You need a lot of data to get it to converge, I think.
Any more questions?
Albert on Slack would like to know what extent are data processed on the fly?
At T-REXX, so at T-REXX, as soon as you start a chip, we start processing. So we process on the fly for you. And as I said, you get your initial data processing stats a couple of minutes after the data collection completes. And then we run into the dimple pipeline and take you to electron density. So we're not absolutely real time, but pretty close.
Maybe one question from me. In terms of you showing this, there's always a large energy barrier for someone to start such an experiment. And one wonders whether the whole time resolve thing is a bit of an oversell. How easy is it to get into it?
So I would always start with trapping. All right. And if you can answer your science question with trapping experiments, do that. Okay. If you then, I think we're with the work that's being done at T-REXX with I-24 VMXI here in Diamond, but also now ID29 and Micromax coming online. The beamline staff are putting a huge amount of work into making those beamlines user-friendly. You still need to put the legwork in on getting your samples ready, okay? And you still need to put the legwork in interpreting your data, but the experiment itself, I think, is now becoming much more accessible than it was before.
So one more question on Slack.
Bill Shepherd wants to know how many or which beamlines are using microfluidics or jets or fixed targets?
Right, okay, recent reviews. So we're mostly using fixed targets and occasionally microfluidics. I think at Swiss Light Source, they've been using the viscous jet a lot. And I think also you've been using that at I-24 viscous jets.
Yeah, we have the viscous jet, but it's not as requested. So we use the fixed targets a lot more. But if you want to do
exactly
viscous jet,
where you run into problem is the free jets, because the crystal velocities are just too high, and they basically transition across the X-ray beam very fast. So they go in a microfluidics experiment, they're traveling across in maybe sort of 70 to 100 microseconds, unless you really dial the flow back. And if they're in a free jet, they're going much faster. And that's really what limits the free jets on the synchrotrons. But with the machine upgrades, we may well get to a point where they also become a tractable mount.
And there's also VMXI with the tape drive.
VMXI with the tape drive as well. Yes, thanks, Sam.
Okay, thank you very much. Fantastic.